Sneeze of the second half of the semester. Actually, we're way past half, which is good. There's 6 weeks left, so it really, if there was ever a time to lock in, it's now. But understand that you've done some of the photosynthesis stuff in your lab already, which is good. So hopefully this will elaborate on some of the stuff you did with the pigments, and that will make a little more sense. If it didn't make sense the first time, maybe it already made sense. But I did want to mention a couple things about the exam. As always, some people ask about strategies and things, and I do hope you don't think I'm ever withholding things that would help you do well on the exam. It's in my best interest that you do well on the exam. So I have the responsibility of asking you questions to make sure you know stuff, but certainly if you do well on that exam, that reflects well on me too, and that makes me happy and makes me feel like I'm doing something worthwhile. So I'm not withholding strategies. One thing I would suggest though, and I think this may be more pertinent than it would have been in my generation when I was a young lad learning biology, there just weren't as many distractions. And I think that you guys are probably better adapted to handle distractions because you were born with the cell phone inside your body. And so you've never been without technology. But one thing I might encourage you to do as an experiment, since you are scientists and you can understand the idea of trying something to see if it works. What I would suggest as an experiment for the next exam is see if you can detach yourself for like extended study periods from your technology. Instead of using a phone and an iPad and a computer to simultaneously bathe yourself in Quizlets and slides and digital images and videos, just give your brain a break so that all it's thinking about is just what's on a piece of paper. Drawing, especially the photosynthesis, is so amenable to this. Drawing pathways. This thing leads to this. Writing out lists. Drawing the anatomy of the leaf. Things that are inherently paper-driven. And don't rely on just looking at things and having this digital interaction, but just take a breath. Then, when you're done, take your cell phone, put it on the ground, and then crush it under the heel of your boot. And you'll be so much happier in this life. I know that's not reasonable or possible, but I would, the first half of that was completely serious. To try and detach yourself from all of the things that are just making your brain go haywire. Just slow down and try some paper studying that is devoid of distractions and multimedia. I know that stuff can help. I know it's sometimes useful. I know sometimes it's the best way to understand complex biological things where watching a video or seeing it in motion helps. I know that. I'm not naive about that. But sometimes the distractions, like I'll hear a student say, I've been out here studying for 2 hours, and I see them because they're at the table in the hallway where people are going back and forth, and they've got TikTok on and they have the computer on, so there's a sliver of their brain that's studying, but so much of it is just absorbing this, experience of life. And so if you can find a quiet place and really just focus on a single thing, I think that would make a difference. And if any of you decide to run that experiment, I would like to know how it goes. Well, I'll know how it goes. I just won't know if you've run the experiment or not, because I'll see your grades, obviously. All right, well, let's try something new and refreshing. Let's think about how we are getting our oxygen. I brought a friend of mine. This is Janice Jade. She is the descendant of one of your ancestors. A student who worked for me as a research student in 2015, maybe 16-ish, and now she is a family physician in Louisville. But before she graduated, she gave me a tiny jade plant that was this big. And this is the granddaughter of that jade plant, because the real jade plant is now like this big in my house, and I pinched off a leaf and grew a little baby. And so it's the circle of life. I'll tilt it this way so she can see you all. I feel like Jalen is here with us, but instead she's in Louisville making 8 times my salary. Do I deserve any of that? No, I don't. I don't at all. I just got the plant, and that's fine. That's all I want. Okay. Anywho, takes a moment to get back after break, doesn't it? Before break, we were thinking about how we use glucose and oxygen to produce energy in the form of ATP. And the result of that is that the glucose breaks down, right? And it leaves as carbon dioxide. And one of the byproducts is water. And to remind you of why we end up with water, the final electron acceptor in aerobic respiration is oxygen. And when oxygen is joined by hydrogen ions, as well as the electrons, which have spent their energy producing ATP down the electron transport chain or the oxidative phosphorylation process, when those electrons and those hydrogen ions and the final electron acceptor, oxygen, get together, they form water. Now, I'm sure it's not lost on you that as we think about photosynthesis, we're thinking about the opposite reaction. So this is aerobic respiration. And if we were to reverse this reaction and use carbon dioxide and water and energy to produce organic molecules like glucose with the byproduct of oxygen, that reverse reaction is photosynthesis. So everything that we've talked about so far, is related to what we're going into now, but we can't just go in reverse. And so we have to understand how can we make a complex organic molecule, which requires a ton of energy, and how can we produce oxygen in the process, and how is carbon dioxide going to go from a gas to this solid product that can be used by plants, and then of course we benefit because we either eat the plants, we won't eat Janice, of course. We either eat the plants or we eat the things that eat the plants, right? So these plants are the primary producers, and we are going to be consumers in this process. So we're going to try and understand how these things can lead to this. Now, one thing that is working in the plant's favor, of course, is light. Light energy can come from different sources. Primarily, of course, it comes from the sun. If you've seen the new movie, is it Operation Hail Mary? Is that what it's called?
Project.
What is it? Project Hail Mary. You know that the sun will be gone soon. So there's that. But assuming that the sun does not die, we will have this intense source of energy, right? So the plant is not just gathering ATP from the environment, it is using light and converting it to energy, and that's something we cannot do, and I'll talk about why they can do it and we can't as we go along. Okay, so one thing that we just barely talked about earlier in the semester was the anatomy of plant cells. So now we need to revisit that because we're going to be talking specifically about how photosynthesis occurs in that structure, and so we have to have some of this vocabulary revisited. One thing, if we take a cross-section of a leaf, okay, so the outside of the leaf, just like the outside of our bodies, our integument, consists of epidermal cells. And we won't be thinking about the epidermal cells too much in regards to photosynthesis, but one thing that the epidermal cells are involved with is the transport of gas in and out of the plant. And so the openings where the gas goes through the epidermal cells are called stomata, which is plural. And so since CO2 is coming in and O2 is coming out, we would say this allows gas exchange. One thing to keep in mind too is that and this is easy to lose track of, we do not do photosynthesis, but we do use mitochondria to produce ATP from the oxygen and the sugar that we consume and intake. In the case of plants, it's easy to let your mind draw the conclusion that they are doing photosynthesis, but they're not like us, they're not doing respiration, and that's a false conclusion. Not all of the the energy and the oxygen and the organic molecules that plants produce in photosynthesis are made to be given away, right? As far as the plants are concerned, none of it's made to be given away, right? They're not trying to be eaten. But they do also undergo aerobic respiration. So they use some of the energy for themselves, but they produce in excess. And so when we say gas exchange, it's not just oxygen only going out and CO2 only coming in. They're going to use some of their own oxygen, and they're going to produce some of their own carbon dioxide. They're just going to produce an excess of the organic molecules in O2 that we take advantage of. Okay, inside of the cross-section of the leaf, where we have the actual business of the where the photosynthesis is taking place, these are called the mesophyll cells, or sometimes people say it mesophyll, although I've learned it as mesophyll. But that is the location where we are going to be spending most of our time in this unit. It's one L, right? No, it's two L's. So this is the primary photosynthetic hotspot. And if we dive in a little deeper, we're going to see the chloroplasts. So the chloroplasts, I believe, did you see chloroplasts in the lab? Didn't you watch them in little aquatic plant cells? When you did the microscope, they would have been Elodea cells, I think.
Oh, yeah.
And they were like moving around. Does it ring a bell? Okay. You do have lab, right? I'm a little groggy after the break. It's okay. I am too. But the little chloroplasts you saw, if we were to pull one out of the cell and slice open a little pie slice out of it, this is what we would see. And it's hard to see when it's in the cross section. Let's see. This is what it would look like this is what it looked like when you saw it under the microscope. But if we were able to go in closer to an actual chloroplast, and one thing that you didn't have the luxury of in the lab would be to see the cross section. And you can actually embed cellular structures like a chloroplast or like mitochondria or ER, etc. You can embed them in a solid substance. A lot of times people use a type of wax, and then you can do slices through them so that you can actually see something like this. And what we're looking at here can relate back to this. And so the little stacks, well, let's back up. So some things that we know about the chloroplast. Bless you. One is that it has, this is not going to relate to photosynthesis, but we do want to remember this, is that it contains its own DNA. So that makes it similar to the mitochondria, which also contain their own DNA. And incidentally, if you should ever participate in a crime scene investigation or be perhaps the reason the crime scene investigation is happening, be aware that hair is not a great forensic tool. Hair, usually, if it's broken off on the shaft, contains only mitochondrial DNA because most of those cells are dead and the nuclear DNA actually breaks down. If you have a hair follicle, that is useful because the hair follicle has cells that have nuclear DNA. But just a little FYI, that was a really popular thing in the early forensic medicine shows. People would find a hair and they would identify the perpetrator, and that's almost impossible to do. But I digress. So it contains some DNA. Also, like mitochondria, it has an outer and an inner membrane. Actually, it contains is probably not a great word for membrane. Let's say, we just say has two membranes. And in between those membranes is the intermembrane space, just like in mitochondria. Now, most of the action, though, in photosynthesis is going to take place inside the intermembrane. So the intermembrane space plays a different role here than it did in aerobic respiration. So inside the intermembrane space, we have a few different things. We have the stroma, which is the fluid that surrounds thylakoids. And we'll think of the stroma kind of as a location too. So we'll say things are moving in the stroma or things are, events are occurring in the stroma. So we'll talk about it as though it was a location, but it's also a fluid-filled space. In fact, everything in a cell really is a fluid-filled space. And then the most important part, well, I shouldn't say most important part, the part we will spend most of our time on are the thylakoids. And the thylakoids are going to be the site of pigments and most photosynthetic reactions, and abbreviate reactions, RXN. Now, anytime we have a membrane-bound structure in a cell, especially in an organelle, the inner compartment of that is often called a lumen. So, for example, with the endoplasmic reticulum, there is an ER lumen, which is on the inside of the membrane of the endoplasmic reticulum. And so the thylakoid is similar in that respect. So the inner component is the lumen, and we'll be distinguishing that from the outer side of the thylakoid, which would be the stroma. And so let's put that as a subcategory in our little list of anatomical places. So the lumen is the inner compartment within the thylakoid membrane. And then our last little anatomical detail, I think, we need to talk about, unless I'm forgetting one, is the term granum, which is defined as a stack of thylakoids. So they don't actually exist as little independent units. They exist in these little stacks that look like stacks of mints or coins. And the plural of granum, which we will sometimes use, is grana. Okay, anything I'm forgetting? I don't think so. Okay, so when we talked about aerobic respiration, we mentioned that, oh yes. Oh, it's an abbreviation for reactions, RXN. So it actually isn't a word, it was an abbreviation. Okay. Sorry. What was I gonna say? Let's see how easy it is to throw me off track. Just say, there's a bug on the wall, Dr. Payton, and I'd be done. Oh, I know what I was saying. In aerobic respiration, things are compartmentalized. So things are happening in certain places for certain reasons. And that's gonna be echoed here in photosynthesis, and this is really important because it has to do with the location of the pigments and whether or not light is involved. So this is more than just a passing matter. We want to really remember and focus on this. So we're going to break photosynthesis down into two major components. The first major component will be called collectively the light reactions, okay? And the second major component will be called the Calvin cycle. And the light reactions and the Calvin cycle are passing molecules between them. So one is producing something that the other needs, all right? Now, when the light reactions occur, this has to happen, as the name implies, in the presence of light. Whether it's artificial light or sunlight, it has to happen in the presence of light. But the Calvin cycle does not. Some people use the term light-independent reactions. If you've learned photosynthesis before in another class or high school or whatever, you might have learned it as the light-independent reactions. And that is a reference to the fact that the Calvin cycle does not require light. So if you were a plant, like our little jade plant here, you would need light to do the light reactions, but when the sun goes down and when the office lights shut down, she actually lives in our main office, 103, when the lights are gone and she can no longer do light reactions, she can still do the Calvin cycle. So the Calvin cycle is going to use the components from the light reactions whether there's light there or not. It doesn't have to happen in the dark, but it can, and that makes it unique from the other reactions. Maybe we should say some of this. I can't remember if I say this in a slide later or not. let's go ahead and mention it now. It's importante. Okay, so photosynthesis is going to be broken into two components. The light reactions, and we'll fill in a couple things about that, and then the Calvin cycle. Okay, the light reactions are going to take place only, oh wait, I forgot, I don't want to write things in this no man's zone. Take place only in the presence of light. First of all. And one of the things that the light reactions do is to produce ATP and a molecule that we have met a relative of, NADPH. NADPH should remind you of NADH. Do you remember what we called NADH? It had a particular sort of label. We said it was a blank blank, an electron carrier. And so NADPH is also going to play that role of being an electron carrier. Another thing that happens in the light reactions is the production of oxygen, right? This is a major output of the photosynthetic process that we need and that plants need as well. So it produces ATP, NADPH, and oxygen. Okay, and oxygen, when it's by itself, is always going to exist as oxygen gas, O2. Okay, the Calvin cycle does not require light. So again, some people call them the light-independent reactions. The Calvin cycle is going to be using the ATP and NADPH, which means after those things are used, this is very typical of what we have seen previously, so this hopefully integrates with your previous knowledge. ATP, when it donates energy, breaks down to ADP and inorganic phosphate. NADPH, just like NADH, when it releases its electron, it becomes, instead of NAD+, it becomes NADP+. So the difference there is that it has a phosphorus atom, but otherwise we'll be treating that the same way. So the Calvin cycle, since these are breakdown products, I'm going to use the term releases instead of produces. Well, no, maybe I should use produce. Let me be consistent. That's important. So it produces ADP, NADP+, and this is important, of course, it produces the precursors to make sugars. Now, one little side note, let me write this first. In ADP, NADP+, and sugar, well, let's be more scientific. Let's say carbohydrate. And carbohydrate precursors. The carbohydrate precursors, the reason I use that expression instead of saying glucose, because in our original reaction we said, glucose breaks down and then photosynthesis makes glucose and we have this beautiful cycle. But not all of the carbohydrate precursors go to become glucose. If the plant needs structural carbohydrates, those precursors might be used to make cellulose. The plant precursors might be used to make other sugars that the plant needs, like sucrose, right? A lot of plants produce sucrose. And so some of the carbohydrate precursors become glucose, but not all of them. So let's be accurate about that. Okay, and then the last thing that the Calvin cycle does, which I'll put over here because we've run out of room there, is that it takes CO2 from the environment and it incorporates it into what will eventually become these carbohydrates. And so that process is called carbon fixation. So we're going to say performs carbon fixation And then to define carbon fixation, this is the conversion of CO2 from the environment into carbohydrates. I wish I had brought my drink, my non-energy drink. All right, anything else about this that we want to know? Let's see. Carbohydrates produce ATP. All of this is happening in the chloroplast. So the light reactions, I guess I didn't put the location. Light reactions are taking place in the thylakoids. And the Calvin cycle takes place in the stroma. All right, a lot of good information already. How are we feeling? Who likes oxygen? Oh, come on. Okay, let's be honest. Who likes jade? Thank you. She needed that. You spent all spring break alone in the office on the windowsill, longing to be outside. Dr. Lydiard, my extremely tall boss, was telling me about a new book he's reading. When I went in to get Jade, I talked to him for a minute. And he's reading a book about plants and how there may be more going on than we know. And they created a experimental, well, you can genetically engineer plants to make fluorescent proteins. And those fluorescent proteins can be made in response to cellular changes. And the experiment he told me about in the short 2 minutes that we were chatting was that they cut off a piece of a leaf of a plant, and the assumption was when you do that, that little piece of leaf or that little part of the plant will respond and heal over or whatever. But what happened that was unexpected was, I'm waiting for the train to leave the hallway. What hallway are they in? It's so long. What happened instead was when you cut the leaf on one side of the tree, the whole tree started making these fluorescent proteins in response to this small injury. In other words, it was not an isolated injury. It's like when you prick your finger on a needle or a thorn or something, you feel it throughout your body. Your brain senses it, your hand senses it. And so the idea that plants are just, a clump of cells without any sort of, connectivity between them apparently is false, right? Yep. Okay, let's think about light. So, of course, we have to dabble in physics just a tiny bit, not too much, but there are a couple of graphs that I think you worked on in the lab. But have you been tested on this yet? Has this exam in the lab taken place where you talk about the photosynthetic graphs?
No.
Okay, good. That's good. So we can hit that topic. I was afraid I'd missed my chance. That would have been on me. All right, so a couple things about light energy. We don't need to understand everything about the bizarre physical nature of light. But one thing that we do want to understand is that light's energy and what we perceive as color can be depicted also as wavelengths. And those wavelengths have different lengths, and when we say a wavelength, we're talking about the distance from a peak to a peak. And so this can be in the nanometer range, it can be in the centimeter range, it can be in the meter range, depending on what type of electromagnetic radiation we're talking about. In the case of photosynthesis, what we're thinking about mostly will be found in the visible light spectrum, which is longer wavelength than ultraviolet light, but less wavelength than infrared light, two types of radiation that you're no doubt familiar with and are exposed to different degrees. But in the visible light spectrum, this is what we can actually perceive. So 380 nanometers to 740 nanometers. When you use the spectrophotometer in the lab class, you are adjusting the light source so that it is emitting light at different wavelengths, and then you're testing to see how the photosynthetic process might respond by looking at how pigments absorb light. Okay, and so pigments are any chemical that absorbs light. So you make pigments, for example, melanin is the pigment, the primary mammalian pigment that you make. If you produce eumelanin, that's the very dark form of melanin. So if you have black hair, or if you tan darkly, or have dark skin, you're producing the dark form of eumelanin. If you have fair skin or red hair, you're producing the light form, which is called pheomelanin. But that's a pigment. It absorbs light. In the case of our cells, and mammalian, and actually all vertebrates for that matter, as well as fungi, do insects make melanin? I don't know. In any case, doesn't matter. The reason that it exists for us is that it protects our cells' DNA. And so the melanin absorbs light, especially UV light that would otherwise hurt our DNA. So it serves a purpose. In the case of plants, though, they are absorbing light in the visible spectrum. And it doesn't absorb evenly. And that's the point of these, the graphs we're going to look at. Okay, now to mention for a moment the difference between us and them, and by them I mean the plants, is what you can do with light energy. So back in the early days when we had just started this class, we just met, we looked at the electron, the levels of electron energy that existed around a nucleus, and we talked about how electrons exist in somewhat predictable places. Now, all of this, of course, is a gross oversimplification of what's really happening, but they exist in roughly predictable places, and those electron shells contain certain amounts of energy. So if you were an electron that was close to the nucleus, you would have less energy than if you were an electron that was farther from the nucleus. So if you're in some of these larger atoms that have multiple electron shells, the further you get away from the nucleus, the more energy those electrons are going to have. Now, the electrons that we're going to be thinking about are existing in two energy states. Okay, and so the normal state, this electron is in its so-called ground state. That's its default energy level, and it's existing a certain distance from the nucleus. Photons, which are units of light energy, I should write some of this down, I guess, are going to strike the atom, and that light energy can be absorbed by the electron. And remember, an interesting thing about electrons that was one of the main points of aerobic respiration is that electrons can exist at different energy levels. So we had high energy electrons that were passed from enzyme to enzyme by electron carriers, and they were allowing those enzymes to do work, which was to pump protons across the membrane of the mitochondria. Okay, and so electrons don't just exist at one flat-lined energy level. And so it should not surprise you then if we think about photosynthesis, when we have photons, which are units of light energy. These units of light energy can energize an electron to jump into a different energy state, which we call a photoexcited state. Okay, and so one way you can think of this similar to a chemical reaction, although it's not technically a chemical reaction, is that if we add light energy to an electron in the form of photons, and the reason we say photons is that you can measure, you can quantify it. So you can, for example, when people do experiments in photosynthesis to try and understand this process, you can, what's the right word? You can control the amount of light energy that is given. That way you can have measurable outcomes. You can predict how much light energy does it take to make X amount of oxygen, for example. And so photons is a unit of light, just like centimeters is a unit of length, and so forth. All right, so we add light energy to the electron, And then that electron becomes a photo-excited electron. Okay, and it's sort of redundant to say this is in its excited state, but an electron can be in an excited state for more than one reason. So we'll say photo-excited to specify it's because of a photon, but an excited state can occur in an electron from other energy sources as well. Okay, and then when that excited state goes back to the ground state, energy will have to be released. All right, so you can invest energy, get your electrons excited, then you can release energy and the electrons go back to where they were. All right, now the difference between us and Jade, Janice Jade, at that point, JJ, the difference between us and her is that when her electrons are photo-excited, she can harness the energy that is released. All right, so for plants, this released energy can do work. For us, it cannot. So we can be photo-excited. In fact, you look pretty photo-excited right now. Not so much. Not feeling it, huh? Does that Red Bull create a photo-excited state? Or just frenetic energy? Oh, don't. Close your eyes, Jade. You see that behavior. So when you go outside, your electrons will be photo-excited. The difference is you can't do anything with it. And that is no flaw of yours. That is just how we're made. And so when we go back to the ground state, we release energy in the form of heat, but it's not usable. Okay? That's why we need the plants. They can do things we cannot do. Now, one of the reasons they can do that is because they have different pigments than what we have. So you've probably seen this image and probably that image in your lab when you talked about the different pigments and their different absorption spectra. And so chlorophyll A and chlorophyll B are the main pigments that we're going to be thinking about, but we'll think about beta carotene too as an auxiliary pigment. And these, of course, are by no means the only three pigments in plants. These are just the main ones that we're going to talk about. But the more pigments that a plant has, the more opportunity it has to absorb light at different wavelengths. So it's in a plant's best interest to have a variety of pigments, although many do not. And so the pigments that we will think about are chlorophyll A and B. Is it a little A? And then beta carotene. And the pigments can do something for plants, and they can sometimes do things for us as well. So for example, why do we want beta carotene in our diet? What is the benefit of that? It can make us orange. If you eat enough beta carotene, you will turn orange. This is not a joke. You really can turn orange. Some people do that on purpose. Why not? It's a good look. Apparently it was a popular thing in Europe to eat a lot of beta carotene and turn a sort of ruddy orange color. I'll be surprised if I've not already told you the story of my friend's carrot baby. Did we not talk about the carrot baby? I had a friend who was following a very strange religious diet. I'm not sure where the intersection of diet and religion should be. When it comes to carrots, at least. I understand meat and stuff. I get it. But carrots? Anyway, the baby was fed so many carrots. When you're a baby, you don't get to say no more carrots, right? You just cry, and then they think, oh, you're hungry, more carrots. So it's a tough situation to be in, and the baby started to turn orange. And, you know, I'm not proud of this, but behind their backs, we called it the carrot baby. But I wasn't proud of what they were doing to the baby either. But I don't think it was unhealthy. It's just weird do you not notice when your baby is turning colors? And say, maybe we've gone too far? Maybe this is a sign to show some moderation, which is also a basic tenet of most religions, moderation. Anyway, point is, it's healthy. Just use a little self-restraint. Unless you want to be orange, and then go for it, man. Go for it. If one of you came in orange the next class period because of carrot consumption, I would be just over the moon. Like taking science to the next level.
I wonder how many carrots you have to eat though.
I don't know. Don't do this really. Someone's going to get sick and then I'm going to die.
Could we get extra credit if we come in
Get-
orange?
Yeah, if we all if we all like show up
Please,
orange, will you give us
don't
extra
really do
credit?
this experiment. Understand where the joke ends. But that wasn't where I was headed with that. But I can't see beta carotene and not think of carrot baby. But why do we need beta carotene? There's a reason, besides our appearance.
Oh, your eyesight.
Vision, yes, it's an important precursor to molecules that you need for light absorption in your eyes. So you have to have some of this in your diet, or your vision will suffer. Okay, so what do we know about the chlorophylls and the beta carotene, which is part of the larger family of carotenoids? So it's not the only carotenoid. But one thing that you should notice about this is some structures that I hope are a little bit familiar to you, like this long chain of carbons and hydrogens, and over here this long chain of carbons and hydrogens. And those are going to be very hydrophobic, right? Because they look similar to fatty acid tails, right? A fatty acid tail wouldn't have all these little side methyl groups, but it's the same atoms, right? Carbons and hydrogens in this long chain. And so those are going to be very hydrophobic, and that allows these things to interact, for example, among other things, with membranes. But the other thing you might notice is up here, you've got some molecules that are going to give a little bit of a polar aspect to this molecule. And I think this, if memory serves me, a long time ago, one of the questions about why the pigments migrate differently on your chromatography sheet was about their polar groups. Does this ring a bell? Like, why does chlorophyll A migrate differently than chlorophyll B?
No.
Well, it may not be a question anymore on that. I remember when I taught the lab years ago, that was a question. Why do they migrate differently? And the reason they migrate differently is they have different degrees of polar groups. And so chlorophyll A, for example, is the one depicted here. And so it has an oxygen there, an oxygen there, an oxygen there, an oxygen there. And is that it? 1, 2, 3. It has 4 oxygens. And those oxygens notoriously add polar characteristics to the molecules where they reside. Chlorophyll B, on the other hand, is just like this, except instead of this CH3 group, which is a methyl group, it has a carbon, an oxygen, and a hydrogen. And so that means it has 5 polar groups. So let's note that so I don't have to count them later. And when you do the chromatography, or when you did the chromatography, the polarity of the molecule is what allows it to migrate differently up the length of that paper, the chromatography paper. And so chlorophyll A and B, with their different number of polar groups, but otherwise exact same structures, will actually migrate differently. That single oxygen is enough to set it apart. Okay, now